Hi all, today we are dealing with another topic, that is Hybridoma technology and monoclonal antibody production. These are the topics that will be discussed today. Monoclonal antibodies versus polyclonal antibodies, principle of a hybridoma technology, advantages of a monoclonal antibodies, steps involved in the production of monoclonal antibodies, types of monoclonal antibodies, approved therapeutic antibodies, applications of monoclonal antibodies, side effects of monoclonal antibodies. Getting into the introduction, it is a technique to generate monoclonal antibodies, method for producing large number of monoclonal antibodies generated by a single clone of plasma cells or same type of a clone are called monoclonal antibodies. Monoclonal antibodies can recognize a single epitope. Hybridoma technology was discovered in 1975 by two scientists, George Kohler of West Germany and Cesar Milstein of Argentina, for which they received the Nobel Prize in 1984 in Physiology and Medicine. Monoclonal antibodies are nowadays used in the treatment of AIDS. Monoclonal antibody versus polyclonal antibodies. Monoclonal antibodies are the antibodies that are identical because they were produced by one type of immune cells, B cells, clones of single parent cell bind to a single specific antigen. In case of a polyclonal or conventional antibodies, those are the antibodies from multiple clones of B cells and therefore bind to a number of different epitopes of antigens. Production is expensive in case of monoclonal antibodies, and in terms of polyclonal antibodies, their production are inexpensive. Long production time are required for monoclonal antibodies, and on the other hand, rapid production in case of polyclonal antibodies. Large quantities of specific antibodies can be produced through monoclonal antibody. Large quantities of non-specific antibodies can be produced through polyclonal antibody production. Recognize a single epitope on an antigen is the specialty of monoclonal antibodies. In terms of polyclonal antibodies, they can only recognize multiple epitopes on an antigen. Production is continuous and uniform for monoclonal antibodies, while the different batches vary in composition in case of polyclonal antibodies. Hybridoma technology's principle is clone of B cell is stimulated against a single epitope of antigen is fused with an immortal cell, example myeloma cell, capable of multiplying indefinitely to produce a hybridoma cell. This hybridoma cell has two unique properties. It produces monoclonal antibodies of the same antigen specificity. Second, multiplies indefinitely, producing a clone of identical cells. Advantages are monoclonal antibodies have very specific target. Monoclonal antibodies do not have intercellular activity. As a result, there are few anticipated side effects and reactions. Monoclonal antibodies are not metabolized by kidney or liver but are catabolized within the cells, resulting in amino acids which are recycled within the body. Steps in the production of monoclonal antibodies. There are basic 10 steps. First, immunization of a specific animal. Second, screening of mice for antibody production. Third, preparation of myeloma cells. Fourth, fusion of myeloma cells with immune spleen cells. Fifth, selection of hybridomas. Sixth, screening of products. Seventh, cloning and propagation. Eighth, production of monoclonal antibodies. Ninth, purification of monoclonal antibody. Tenth, characterization and storage. First, immunization of a specific animal. Experimental animals are immunized 2 to 3 times with antigen intravenously. An antigen that is prepared for injection either by emulsifying the antigen with a France adjuvant or other adjuvants. Screening of mice for antibody production. Blood samples are drawn from mice for measurement of serum antibodies with various techniques such as ELISA and flow cytometry. Preparation of myeloma cells. Myeloma cells are mutated and immortalized cells. These cells are cultivated in 8-asaguanine a week before cell fusion. A gene which is involved in the synthesis of DNA is HGPRT, hypoxanthine guanine phosphoribosyl transferase. This gene is made non-functional through mutation. These cells are not capable of producing neither antibodies nor a HGPRT enzyme. Fusion of myeloma cells with immunospleen cells. 5 different types of cells are formed. Hybridoma cells, fused B cells, fused myeloma cells, unfused B cells, unfused myeloma cells. Hat medium before fusion and after fusion. The scenario of the cells is as shown in this diagram. Selection of hybridomas based on inhibiting the nucleotide synthesizing machinery. Mammalian cells can synthesize nucleotides by 2 pathways, de novo pathway and salvage pathway. When cells deficient in HGPRT are grown in a medium containing hypoxanthine, amnocterin and thymidine, they cannot survive due to inhibition of de novo synthesis of purine nucleotides. Thus, cells lacking HGPRT will die. Unfused normal spleen cells and fused spleen cells cannot grow because of their limited lifespan. Only hybridoma cells possess the ability of myeloma cells to grow in vitro with a functional HGPRT gene obtained from lymphocytes. Thus, only hybridoma cells proliferate in hat medium. Selection of hybridomas. When the cells are cultured in a hat medium, only the hybridoma cells grow while the rest will slowly disappear. Screening the products. The products are screened using ELISA and RIA. Cloning and propagation done by the limiting dilution method and soft agar method. Production of monoclonal antibodies. There are 2 methods of production. In vitro tissue culture technique, where we culture the cells in a laboratory. Disadvantage is the production of monoclonal antibodies is low in the culture bottles. Then the second method is in vitro animal method. Here yield can be increased by growing the hybrid cells as sites in the peritoneal cavity of mice. Disadvantages are a collection of monoclonal antibodies from ascitic fluid is associated with the heavy risk of contamination by pathogenic organisms of the animal. Purification of monoclonal antibodies were done using various methods such as harvest, centrifugation, filtration, protein A chromatography, low acid hold for a viral inactivation, viral infiltration, filtration, ultrafiltration, etc. Characterization and storage. The stability of the cell lines and the monoclonal antibodies are important. For that, their storage is really important. It is stored in liquid nitrogen at minus 196 degrees centigrade for several stages of cloning and culture. Monoclonal antibody types are of 4 types: murine, chimeric, humanized, fully human. Approved therapeutic antibodies is shown in this diagram. Then the monoclonal antibodies application, diagnostic application, biochemical analysis, diagnostic imaging, therapeutic application, direct use of monoclonal antibodies as the therapeutic agents, monoclonal antibodies as targeting agents, diagnostic applications, biochemical analysis for the pregnancy, cancers, hormonal disorders, infectious diseases. In case of the diagnostic application, diagnostic imaging, also known as immunoscintigraphy, myocardial infarction, deep vein thrombosis, atherosclerosis, bacterial infections, cancers, tumor markers and associated cancers are shown in a tabular format. Then the therapeutic applications, the direct use of monoclonal antibodies as therapeutic agents and monoclonal antibodies as targeting agents. Direct use as therapeutic agents in destroying disease-causing organisms, in the treatment of cancer, in the immunosuppression of organ transplantation, the treatment of AIDS, in the treatment of autoimmune diseases. Monoclonal antibodies as targeting agents in drug delivery to target specific cells in a disease treatment, in the dissolution of blood clot used as immunosuppressants. Side effects of monoclonal antibodies can be seen: headache, weakness, fever, cough, muscle ache, chest discomfort. Production of monoclonal antibodies is shown in a diagram as follows. Firstly, immunization of mouse, then cells are fused, selection of hybridomas in hat medium, isolation of hybridomas of a single specificity, screening of products, cloning and propagation, characterization and storage. These are the references, and thank you.